Hello friends, I myself Dr. Bhagwan Mali, Department of Botany, Tulzaram Chaturchanda College, Paramati. My friends, as you know that we have been dealing with the plant biotechnology paper from your TyBSc Botany. From that, unit third is there germplasm conservation and cryopreservation. We have discussed at length whenever the colleges were offline regarding what is germplasm, what is necessity of conservation of all these things, what is cryopreservation, but in detail today we have to get idea. My friends, germplasm is nothing but the hereditary material transmitted to the offspring through the germ cell. Hereditary material which is transmitted to the offspring through the germ cell. In the sense, my friends, एका पिढीतून दुसऱ्या पिढीमध्ये जे हेरिडेटरी मटेरियल ट्रान्समिट होत आहे जर्म सेलच्या द्वारे त्याला म्हणायचं जर्म प्लाझम. Actually, germplasm provides the raw material for the breeder to develop the various crops. We know that just to provide three basic needs, food, shelter, and clothes to the over increasing population, we'll have to produce breeding varieties, and for that one, that material, that is germplasm, very having a lot of importance. Thus, the conservation of a germplasm assumes significance in all breeding programs, isn't it? Whenever we are interested just to develop a breeding variety, at that time we'll have to definitely deal with the germplasm. The very objective of germplasm conservation is to preserve the genetic diversity of a particular plant or genetic stock for its use at any time in future. In recent years, many new plant species with the desired and improved characteristics have started of replacing the primitive and conventionally used agriculture of the plants. As you know that we are going to develop very desired and improved characteristic and new varieties, but primitive varieties are and conventional varieties are replaced by them, replaced by newly developed improved characteristic having plants. It is important to conserve the endangered plants, or else some of the valuable genetic traits present in the primitive plants may be lost. My friends, for your kind information, in the last lectures of our offline lectures, I have given you an idea about RET. R stands for RET plants, means R stands for rare plants, E stands for endangered plant, and T stands for threatened plants. We have discussed at length about all these things. So it is necessary to conserve the endangered plants because they are having a very interesting characteristics with them, traits with them. For that one, we'll have to preserve. The global body, namely International Board of a Plant Genetic Resources, has been established for the germplasm conservation. International Board of a Plant Genetic Resources. International Board of a Plant Genetic Resources, PGR. Plant genetic resource has been established for the germplasm conservation. Its main objective is to provide necessary support for the collection, conservation, and utilization of plant genetic resources throughout the world. My friends, you'll have to remember this International Board of a Plant Genetic Resources, which is established for germplasm conservation. Its main objective is to provide necessary support for the collection, conservation, and utilization of the plant genetic resources throughout the world. My friend, there are two main approaches of germplasm conservation, that is for genetic material, in situ and another is ex-situ. In situ conservation, the conservation of germplasm in their natural environment, that word you will have to keep in mind, in situ. Whenever we are preserving, conserving the germplasm in their natural habitat environment by establishing and biosphere reserves or national parks and sanctuaries regarded as in-situ conservations. But there are some limitations of in-situ conservation. The risk of losing germplasm due to the environmental hazardous may not be that environment suit to the growth of that or conserve the germplasms. That is a hazardous. The cost of maintenance of the large number of genotype is very high. Again, just to provide as per the requirement, the environment to the conserving organism, it is very costly. Ex-situ conservation, it is a cheap method for the preservation of a germplasm obtained from the cultivated and white plant material. The genetic material in the form of a seed or in the form of a in vitro culture plant cells, tissue or organ can be preserved as a gene bank. My friends, ex-situ conservation, that is equal to in laboratory in the form of a gene bank for the long-term storage under suitable conditions. For successful establishment of a gene bank, adequate knowledge of a genetic structure of the plant population and the technique involved in the sampling, regeneration, maintenance, the gene pool, etc. are essential. My friends, ex-situ conservation, although that is a very, we have to say that useful, significant, having a lot of importance in the life of the human being, but also it requires the skilled fellow knowledge of genetic structures, isn't it? Then only it is possible for the long-term storage with the help of a gene bank. Now, advantages of a ex-situ conservation, my friends, as already I have given you idea about all these things, allowing the natural selection to act which cannot be recreated ex-situ. The maintenance of a viable and self-sustainable population of a wild species in their natural state represents the ultimate goal, but habitat destruction is inevitable and endangered species. Ex-situ conservation provides person to study the biology of and understand the threats and endangered species, as to also has an advantages of preserving plant material and making it available for the researchers purposes without damaging the natural population. That is most important. Ex-situ conservation help us just to conserve the plant material in original form, and then we can make available to the scientists for the further research without damaging natural population. Therefore, conservation of ex-situ is a complementary to the in-situ conservation and can act as an insurance policy when the species are threatened in their natural habitats. It is the process of protecting an endangered species of the plants or animals by removing the plant part of the population from the threatened THR threatened habitat and placing it in a new locations, which may be a wild area. My friends, ex-situ conservation has a several purposes. Here, rescue the threatened germplasm. We know that many more organisms are right now threatened for the extinction, and that we can rescue through this. Then produce the material for the conservation biology research. Then bulk up the germplasm for the storage of various forms of a ex-situ facility. Supply the material for various purposes to remove or reduce the pressure from the wild collecting. These are the we have to say that purposes of the ex-situ conservation. Now grow the species with recalcitrant seeds that cannot be maintained in the seed store. Many more seeds can immediately loses their viability. Very small period of their viability is there. In such a cases, we can maintain those of a species. We can store those seeds for long time. Make available material for conservation, education and display. Produce the material for the reinforcement, habitat and restoration. So my friends, these are the way how to say that importance of a ex-situ conservation. Now there is an in vitro method of a germplasm conservation is there that we will see in the next lecture, please.